In this lecture, we are going to discuss chromosomal aberrations. Here, we will mainly focus on the numerical aberrations. Chromosomal aberration is the deviation of the number of the chromosome or the structure of the chromosome from the normal, that is called the chromosomal aberration. A change in the single gene can affect the phenotype of the whole organisms. The change in the gene or change in the chromosome is called the mutation. There are two types of mutation: chromosomal mutation, that is change in the structure or number of the chromosome, and the gene mutation, that is the change in the sequence of the nucleotides at molecular level. Both the type of the mutation result in the change in the modification or in the variation of the phenotype of an organism. Here we will focus on the type of mutation, that is a chromosomal aberration. It is also called chromosomal abnormalities. The numerical aberration will be discussed. Aberration means any deviation from the normal that is usually expected. When any chromosome is deviated from the normal, either in case of the structure or in case of the number, that case is called chromosomal aberration or chromosomal abnormality. Chromosomal abnormality or chromosomal aberration is any change, any abnormality in shape, in size, structure, or number of the chromosome, that is called chromosomal aberration. So we can define chromosomal aberration as the change or abnormality in shape, size, structure, or number of chromosome. Chromosomal aberration reflects an atypical number of or structure in one or more chromosomes. When the number of chromosomes is deviated or structure of chromosome is deviated, the condition is called chromosomal aberration or chromosomal abnormality. Abnormalities or aberration can be in the form of additional material attached to chromosome. When a chromosome is added, when a chromosome having an extra copy of the genes, that is called additional material attachment to the chromosome. It also leads towards abnormality. Chromosomal aberration can be the missing of the chromosome, a part of the chromosome are missed, or the whole chromosome miss. क्रोमोसोम का कोई एक हिस्सा या पूरा क्रोमोसोम मिस होगा, that is the abnormality. Defective formation of chromosome is present but in the defective form, abnormal chromosome, that is the abnormality. So these are three conditions of the chromosomal abnormalities: addition of material, missing of the material, or defective form of the chromosome. Any increase or decrease or any increases or decreases in chromosomal material interfere with normal development and function. Increase or decrease of the chromosomal number also leads towards the chromosomal aberration, and it always interfere with the normal development, normal growth, and normal function of the cells. We mainly classify the chromosomal aberration into two types. Number one, numerical aberration, and number second, structural aberrations. Numerical aberration based on the abnormal number of chromosome. Every organism has specific number of chromosome. If the number of chromosome increases or decreases, the condition is called numerical aberrations. The structural aberration concerned with the abnormal structure of the chromosome. If some part of the chromosome is deleted, or some part of the an extra part is added, or the structure is abnormal, the condition is called structural aberration. So numerical aberration based on the number, and structural aberration based on the structure of the chromosome. In this lecture, we will focus on the numerical aberration. In next lecture, we will discuss the structural aberrations. Numerical aberrations, that is alteration in the number of chromosomes from the diploid set, is called numerical chromosomal aberration. It is also known as the polyploidy. When the number of chromosomes is abnormal, when the number of chromosome is altered, when the number of chromosome is deviated from the normal number of the chromosome present in the cell in an organism, that condition is called numerical aberration. It is also known as polyploidy. These are usually caused by failure of chromosome division, which results in cells with an extra chromosome or deficiency in chromosome. जब डिवीजन के दौरान क्रोमोसोम का नंबर या तो एक्स्ट्रा किसी सेल में मौजूद हो या number of chromosome deficient हो, कम हो, the condition is called numerical aberration. Each species of an organism has specific number of chromosomes. फॉर एग्जांपल ह्यूमन के केस में 23 पेयर ऑफ क्रोमोसोम है। अगर ये 23 पेयर जो 46 क्रोमोसोम होते हैं, अगर ये 46 से एक्सीड करें, 47 में चले जाएं या 45 क्रोमोसोम हो जाएं, both the condition are the abnormal condition. These chromosomes are found in pairs. These always the chromosome present in the cell in the specific number, and all the chromosomes are present in the pair. During gametes formation, the chromosomes number is reduced. During gametes formation, during meiosis, the number of chromosomes, each cell gets half the number of chromosomes. The gametes carry haploid set of chromosomes. During the fusion, during the fertilization, the haploid gamete will fuse, fertilize, and diploid zygote will form. During gametes formation, non-disjunction of chromosome results in abnormality. Non-disjunctions mainly refers to the when the chromosomes will not separate. When separation is not normal, the condition is called non-disjunction. Okay. If either of the aberrant gametes unites with an abnormal one at fertilization, when abnormal gametes will unite with the normal gamete during the fertilization, the zygote will also have an abnormal number of particular chromosome. When the gamete is abnormal, the zygote will abnormal. In some condition, this zygote will not survive. If it survives, then it has different types of developmental problems, different types of disorders that will be found in the next stages or in the developmental stages. Non-disjunction. What is non-disjunction? There are three conditions related to the non-disjunction. Number one, junction. Junction is the association of the chromosome with each other. When chromosomes are arranged at the metaphase plate, in case of the meiosis or the mitosis, this state is called the junction. This is called the junction. Chromosome attach हो रहे हैं एक दूसरे के करीब, this is called the junction. Junction is the attachment or the combination of the chromosome with each other, that is called the junction. And what is the disjunction? Disjunction is the normal separation of chromosomes in meiosis 1 or sister chromatids in meiosis 2. This is called the disjunction. When these chromosomes are separated from each other, जब ये क्रोमोसोम meiosis 1 के दौरान चूंकि होमोलोगस क्रोमोसोम सेपरेट होंगे, this condition is called disjunction. जो जंक्शन बना, अब वो जंक्शन क्या हो गया? वो जंक्शन अब उसने क्रोमोसोम्स को इलादा कर दिया, ये क्रोमोसोम इलादा हो गया और ये क्रोमोसोम इलादा, both the chromosome are separated from each other. This is called the disjunction. This is called the disjunction. meiosis 1 के दौरान होमोलोगस क्रोमोसोम सेपरेट होंगे और meiosis 2 के दौरान सिस्टर क्रोमेटिड्स सेपरेट होंगे. Now what is non-disjunction? Non-disjunction when the separation is not normal. When the separation is not normal, when the chromosome will not separate from each other, either at this stage, at the separation of the homologous, or at the separation of sister chromatids. जब sister chromatids separate नहीं होंगे या homologous chromosome separate नहीं होंगे, this condition is called non-disjunction, and this condition is leads towards the numerical aberration. So non-disjunction is the most important factor in the chromosomal aberration. Non-disjunction, that is a failure of bivalence to separate during meiosis 1. The gametes get both homologous chromosome, both homolog of one chromosome pair. दोनों gametes में एक ही chromosome या homologous chromosome जब transfer होंगे, this condition is called non-disjunction, क्योंकि homologous chromosome separate नहीं हुए. Sometime in meiosis 2, when sister chromatid fails to separate to get two copies of one of the homologs, जब क्रोमोसोम meiosis 2 में जब सिस्टर क्रोमेटिड सेपरेट नहीं होंगे तो दोनों जो सेल्स हैं उनके पास एक ही कॉपीज, जो एक ही किस्म का होमोलोगस क्रोमोसोम ये ट्रांसफर होगा. दिस इज आल्सो कॉल्ड द नॉन डिसजंक्शन. It can also occur during early mitotic division of the zygote. There will be presence of two or more cell lines. Early mitotic division के दौरान भी non disjunction होती है. So non disjunction is the important factor of the chromosomal aberration. meiosis 1 के दौरान जो नॉन डिसजंक्शन है ऑफ होमोलोगस क्रोमोसोम की, हेयर नॉन डिसजंक्शन ऑफ द क्रोमोसोम. क्रोमोसोम ने क्या होना था? क्रोमोसोम ने दोनों सेपरेट होना था. जो होमोलोगस क्रोमोसोम है, वन ईच सेल रिसीव वन पेयर होमोलोगस क्रोमोसोम जस्ट लाइक दिस. बट हियर इज नॉन डिसजंक्शन. ये दोनों जो होमोलोगस क्रोमोसोम है ये एक तरफ ही मूव कर गए हैं. इन द नेक्स्ट सेल क्या होगा? ये जो सेल है इसके पास अब हर एक सेल के पास 3 क्रोमोसोम मौजूद हैं. इसके पास भी 3 क्रोमोसोम मौजूद हैं, लेकिन इसके पास ओनली 1 क्रोमोसोम इज प्रेजेंट। तो यहाँ पे क्या हुआ, यहाँ पे n+1 हुआ, यहाँ पे ऐड क्रोमोसोम ऐड हुआ, यहाँ पे भी ऐड क्रोमोसोम ऐड हुआ, और यहाँ पे n-1 क्रोमोसोम माइनस हो गया। जो पेरेंट सेल उसके पास 2 क्रोमोसोम थे, लेकिन अब जो डॉटर सेल उसके पास n-1, सो नॉन डिसजंक्शन ऑफ होमोलोगस क्रोमोसोम। हियर द नॉन डिसजंक्शन ऑफ सिस्टर क्रोमेटिड। अब सिस्टर क्रोमेटिड जो मेयोसिस 2 में जिन्होंने सेपरेट होना है, अब इन्होंने सेपरेट, ये यहाँ पे सेपरेट नहीं हुई, एक ही सेल में मूव कर गए। जब एक सेल में मूव कर गए तो एक सेल के पास 3 क्रोमोसोम आ गए, दूसरे के पास 1 आ गए। ये 2 सेल नॉर्मल हैं, ये हेप्लॉइड सेल मौजूद हैं। अब यहाँ पे n+1 आ गया, n-1 मीन इसमें एक क्रोमोसोम लेस हो गया, इसमें एक क्रोमोसोम ऐड हो गया। सो टू कंडीशंस मोस्ट इंपॉर्टेंट, नॉन डिसजंक्शन ड्यूरिंग meiosis 1, that is the not separation of the homologous chromosome, and non disjunction during meiosis 2, that is the non disjunction or no separation of the sister chromatids. Both the condition leads towards the abnormal number of chromosome in daughter cells, and both the condition leads towards the chromosomal aberration. There are two types of the chromosomal aberration or ploidy. One is euploidy and second is the aneuploidy. U is for the true and ploidy is for fold. True ploidy, true fold is called the euploidy, and anu, that is called aneuploidy. Mean anu stands for, anu means false and ploidy mean fold. When there is a false fold, that is called the aneuploidy. Both are divided into further different classes. So euploidy और aneuploidy में difference क्या है? euploidy में complete set of chromosome एबनॉर्मल होगा, वो उसकी एडिशन होगी या उसमें एबनॉर्मलिटी होगी। और एनोप्लॉइडी में 1 or more क्रोमोसोम में बी एडेड और डिलीटेड फ्रॉम द whole chromosomes. Here we can classify the numerical aberration into two categories, euploidy and aneuploidy. euploidy में complete set of chromosome ऐड होगा या मिस होगा। aneuploidy में a single chromosome may be added or deleted. euploidy true fold and aneuploidy false fold. euploidy को अगेन हम 3 कैटेगरीज में क्लासिफाई करते हैं, monoploidy that is n, diploidy 2n, and polyploidy 3n. It may be tetraploidy, it may be the pentaploidy further. Polyploidy again divided into 2 categories, autopolyploidy and allopolyploidy. Aneuploidy has 2 types, hyperploidy and hypoploidy. Hyperploidy again 2 types, classified into 2 types, trisomy and tetrasomy, 2n+1 and 2n+2. Hypoploidy, monosomy 2n-1 and nullisomy 2n-2. This is the classification of the numerical aberration. This will be discussed in detail in the further slides. Starting with the euploidy. Euploidy is the variation in the chromosome number that occurs due to increase or decrease of full set of chromosome. इसमें complete set of chromosome increase होगा या decrease होगा, the condition is called euploidy. Complete set mean या तो number of chromosome n haploid होंगे, या diploid होंगे 2n होंगे, या triploid 3n होंगे, या tetraploid 4n होंगे, या pentaploid 5n होंगे. When the complete set of the chromosome is changed, is increases or decreases, this condition is called euploidy. Types of euploidy, there are 3 types, monoploidy, diploidy or diploidy, and polyploidy. Monoploidy is having the half the number of chromosome. Monoploidy have half the number of chromosomes. Gametes are the best example of the monoploidy having half the number of chromosome. Both each gametes receive half the number or reduced number of chromosomes. Drone of honeybee is also having the monoploid or haploid number of chromosome because it is produced as a result of unfertilized egg. Haploid number of chromosome, single set of chromosome, that is called the haploid number of chromosome. These are present in the gametes or drone honeybee. Diploidy is the condition in which the number of chromosomes are in double form or in the double set, 2 set. In most of the plants and animals, the somatic cells contain 2 sets of chromosomes. The condition is called diploidy. Diploidy is formed by the union of 2 gametes during fertilization. During gametes having the half the number of chromosomes, when both the gametes fuse during the fertilization, the haploid condition will become the diploid conditions. This is the diploid number of the chromosome present in the zygote. This condition is achieved by the fusion of the gametes from both the parents, from both the gametes. When both the gametes fuse with each other, then the condition is called zygote, and each zygote having diploid, 2 copies of each chromosome. This condition is called diploid number of chromosome. Polyploidy. Polyploidy. Poly means many and ploidy means fold. When a set of chromosomes are increased from many towards manifold, that is called polyploidy. When a chromosome are more than two, the condition is called polyploidy. Addition of one or more sets of chromosomes to the diploid set results in the polyploidy. When there is more than two sets of the chromosome in an organism, the condition is called polyploidy. It is commonly noticed in plants and rare in animals. Few fishes and amphibians having the condition of polyploidy. A fetus having the polyploidy does not survive. In case of animals, in humans, polyploidy fetus cannot survive. This polyploidy condition can only be tolerated by the plants. This is a condition of the polyploidy, triploidy. When the three set of the chromosome are present, the condition is called triploidy. And when the four set of chromosome are present, then the condition is called tetraploidy. When five sets of chromosome are present, the condition is called pentaploidy, hexaploidy, heptaploidy, octoploidy, and so on. There are two types of the ploidy. Number one, autopolyploidy and the allopolyploidy. Polyploidy has two types: autopolyploidy and allopolyploidy. Autopolyploidy. Auto is the same, and the ploidy, that is the autopolyploidy mean when the manifold are present in the same species and the same organism, that is called autopolyploidy. That is the self doubling of some chromosome set that is in the same organism, from the same organism, that is called the autopolyploidy. Autopolyploids are derived from within a single species. These the chromosome and the polyploid condition is derived from the single species. The condition is called autopolyploidy. Addition of one or more haploid set of its own genome in an organism results in the autopolyploidy. Diploid species in a single organism, when this cell produce tetroid or the four chromosome in its own organisms. ये किस तरह हो सकता है? ये जब क्रोमोसोम अरेंज होंगे मियोसिस 1 के दौरान, मियोसिस 2 के दौरान, जब उनकी सेपरेशन नहीं होगी, तो ईच सेल रिसीव द फोर द नंबर ऑफ क्रोमोसोम, देन दिस टाइप ऑफ सेल विल बिकम द पोलीप्लाइड. एक ही सेल से जब पोलीप्लाइडी कंडीशन ओरिजिनेट हो रही है, दिस इज कॉल्ड द पोलीप्लाइडी कंडीशन. बट ऑटोपोलीप्लाइडी, दिस इज द एग्जांपल ऑफ द टेट्राप्लॉइड. Watermelon, grapes, and banana are autotriploids, whereas apple is an autotetraploid. Different polyploid plants: banana, apple, and potatoes are triploid plants. Cotton is the tetrapolyploid plants, and wheat is the hexaploid, while the strawberries are the octoploid plants. Allopolyploidy. The allo is the product of interspecific hybridization that arises by hybridization between two species. Allopolyploidy increase in one or more haploid set of chromosomes from two different species that result in the allopolyploidy. Triticale is the first man-made cereal. It is best example of allopolyploidy. It is obtained by the crossing a wheat Triticum durum having the diploid chromosome, that is the tetrapolyploidy, that having the 28 chromosomes, and the rye Sicale cereals 2 and 2 set of chromosome having 14. जब इनको क्रॉस किया, इनको जब आपस में हाइब्रिडाइज किया, इससे ट्राइटिकेल एक हाइब्रिड wheat हुई. This is the combination of the chromosome from different organisms and the result in the allopolyploidy. The F1 hybrid that is sometime result in the 21 chromosome. अगर उसमें 21 chromosome आएंगे, that will be the sterile. जब भी polyploidy condition में अगर odd number of chromosome आएंगे, तो the condition is will be the sterile condition. This sterile condition may be converted into the fertile condition by using a chemical or drug that is called colchicine. Colchicine is used for the treatment of the treatment of sterility, for the production of the fertile of organism, which convert chromosome to even number. जो 21 है, ये 21 जब अरेंज होंगे, तो जो colchicine है, ये ड्रग क्या करेगी? स्पिंडल फाइबर को ब्रेक कर देगी और हर ऑर्गेनिज्म में 42 क्रोमोसोम ट्रांसफर होंगे. तो the condition will become the even number and it will become the fertile organism. Allopolyploidy, two species share their chromosomes and result in the heptapolyploid. Species 1 share 4 the number of chromosome in haploid condition, and species 2 share 3 chromosome. When these 4 and 3 having uneven number of chromosome, this having the 4 and the other having the 3, when these two gametes fuse with each other, the zygote having the heptaploid chromosomes, that is the allopolyploidy. This condition comes from two different parents, that's why it is the allopolyploidy. Allopolyploidy, that is a hybridization of two different species A and species B. Species A have 4 number of chromosome in diploid condition, and species B have 6 number of chromosome in diploid condition. Species A undergo the error, that is non-disjunction, and the gametes having 4 chromosome instead of 2. और यहाँ पे 2 क्रोमोसोम चाहिए, चूंकि हैप्लॉइड होना चाहिए, चूंकि ये अनरिड्यूस है, यहाँ पे रिडक्शन नहीं हुई। अब जो गेमीट है इसके पास 4 क्रोमोसोम है। When this gamete fuse or hybridize or fertilize with the parents from other gametes having the normal chromosomes, अब चूंकि इसके पेरेंट्स के पास 6 थे, यहाँ पे 3 नंबर ऑफ क्रोमोसोम्स हैं, हैप्लॉइड हैं। जब ये 4 वाला गेमीट 3 वाले के साथ कंबाइन करेगा, फ्यूज करेगा, जो ऑफ स्प्रिंग होगा दैट इज द हाइब्रिड बट हैविंग द 7 क्रोमोसोम। 4 इससे आए, 3 इससे आए, 3 + 4 = 7। दिस 7 is the hybrid, this is the sterile, क्योंकि इसके पास 8 नंबर ऑफ क्रोमोसोम मौजूद हैं। अब अनरिड्यूस्ड जो गेमीट्स, अगेन इट विल प्रोड्यूस अनरिड्यूस्ड गेमीट्स, इसके बाद जो क्रोमोसोम है वो 7 होंगे। अब ये जो 7 है, व्हेन इट फर्टिलाइज फ्यूज विद द नॉर्मल गेमीट्स हैविंग द 3 नंबर ऑफ क्रोमोसोम हैप्लॉइड, व्हेन इट विल फ्यूज इट विल फॉर्म द 10 chromosomes in diploid condition. Now this have the even number of chromosome, so it is now fertile, viable fertile hybrid will produce, and it is a allopolyploidy. The condition in which the chromosome or the hybrid is formed or the change in the chromosome occur more than 2 chromosome will form from 2 different species, that condition is called allopolyploidy. Polyploids with odd numbers of sets or triploids are sterile होते हैं। फॉर एग्जाम्पल, एक पेरेंट्स में 3 हैं, दूसरे में 5 हैं, या किसी में 7 है, जब ये गेमीट्स बनाएंगे तो ईच शुड रिसीव 2.5, तो इट इज इम्पॉसिबल। दैट्स व्हाई जो आड नंबर वाले क्रोमोसोम होते हैं, आड नंबर पॉलीप्लॉइड होते हैं, दे आर स्टेराइल इन नेचर, बिकॉज होमोलोगस क्रोमोसोम्स पेयर only 2 by 2, and the extra chromosome move randomly to a cell pole, resulting in highly unbalanced non-functional meiotic products. It is for this reason that triploids watermelons are seedless. जो triploids watermelons होते हैं उनमें seed नहीं होते. However, polyploids with an even x number of the chromosome sets can be fertile. जिसमें वो polyploids जिसमें even number of chromosomes होते हैं वो fertile होते हैं if orderly 2 by 2 chromosome pairing occur. अगर उसमें orderly arrangement है, orderly जो pairing है, then the condition will be the fertile. Causes of the polyploidy: somatic doubling during mitosis, के दौरान doubling हुई है तो वो separation नहीं हुई, then the polyploidy. Non-reduction in meiosis 1, in meiosis leading to production of the unreduced gametes, जब gametes में reduction नहीं होगी, each gametes have the same number of the chromosome as in parents, so non-disjunction, polyploidy. Polyspermy, fertilization of egg by 2 male nuclei, and replication of DNA but no cytokinesis. Replication तो हो रही है लेकिन cytokinesis, दो cells नहीं बन रहे. This condition also leads towards the polyploidy. Aneuploidy is the variation that involves 1 or 2 chromosomes within the diploid set of an organism. That the addition or the deletion of 1 or 2 chromosomes within the diploid set of chromosomes is called the aneuploidy. Aneuploidy has 2 types, hypoploidy and hyperploidy. Hypo mean less and ploidy mean fold. जब number of chromosomes are less than the normal, than the diploid, the condition is called hypo. And the hyper, that is the more addition, then the condition when the chromosomes are added from in the diploid cell, the condition is called hyperploidy. Hypoploidy. Hypo, that is decrease in 1 or 2 chromosome from the diploid set, is described as the hypoploidy. Hypo mean less. There are 2 types of the hypoploidy, monosomy and nullisomy. Monosomy. Monosomy is due to loss of a single chromosome from the diploid set. Diploid set से जब एक chromosome loss होगा, the condition is called monosomy. The genetic formula of the monosomy is 2n minus 1. Diploid से एक chromosome reduce किया. Monosomy of autosomes is lethal. Normal diploid cell 2n having the 3 pair of chromosomes A, B, B and C. Monosomic condition में when one chromosome is less 2n minus 1, one chromosome is deleted, C is absent or deleted from this set, that is called the monosomic condition. Double monosomic condition when the lack of one chromosome each from two different pairs. जब दो different pairs से एक set chromosome reduce होगा, that condition is called double monosomic. 2n minus 1 minus 1 genetic formula of the double monosomic. Normal diploid having 2n chromosome having the in diploid condition each chromosome present in the pair. When two chromosomes are missing from two set of chromosome, that is called double monosomic. 2n minus 1 minus 1, A is normal, one B is missing and one C is missing. This condition is called double monosomic. Mean दोनों जो pair of chromosome है उनमें से एक एक chromosome जो है वो loss हुआ. This condition is called double monosomic. An example of monosomy, that's a Turner syndrome, where the individual is born with only one sex of chromosome, that is XO. This is the single monosomic condition. Maize is also the monosomic example of the monosomic. Nullisomy. Nullisomy is the condition in which a pair of homologous chromosome is lost from the diploid set of chromosome. 2n minus 2 is the genetic formula of nullisomy. 2n minus 2. Normal diploid, that is 2n. Each chromosome present in the pair. Nullisomy, two chromosomes are missing. The whole set, that is a C, is missing. This is called the nullisomy. Nullisomy is usually lethal in the diploid and tolerated in the polyploids. This is the nullisomy. The chromosome move to one gamete only, and this gamete is empty, having no chromosome. Both the chromosome move towards this. This is the meiosis 1 non-disjunction. Here each gametes take two pair of chromosome, and this gametes have no chromosomes. This is called the nullisomy. Whole pair of the chromosome is missing. Hyperploidy. Hyper means more, greater. Addition of one or two chromosomes to the diploid set of chromosome results in the hyperploidy. जब एक साथ क्रोमोसोम जब ऐड करेंगे diploid set में, that condition is called hyperploidy. There are two types of hyperploidy: trisomy and tetrasomy. Trisomy results due to the addition of one chromosome to diploid set of chromosome. जब हम एक chromosome ऐड करेंगे diploid set of chromosome में, this condition is called trisomy. Genetic formula is 2n plus 1. Two types of the trisomy. Simple trisomy, increase in number of chromosome in one pair only. 2n plus 1 सिर्फ एक पेयर में इंक्रीज होगा, that is called the simple monosomic condition. Normal diploid condition is 2n each chromosome present in the pairs, and trisomic condition में one chromosome add हो रहा है, 2n plus 1. This C is extra, that's why the condition is called trisomy. Double trisomic condition, the addition of one chromosome in two different pairs. दो different pairs जब chromosome add होंगे, the condition is called double trisomic condition. It is represented by 2n plus 1 plus 1. Normal diploid set, each chromosome pair present in the pair. डबल ट्राइसोमिक कंडीशन में दो क्रोमोसोम डबल होंगे, बी भी डबल हुआ मीन ट्रिपल हुआ, और सी भी नॉर्मली डबल है, अब ये भी ट्रिपल हो गया और ये बी भी ट्रिपल हो गया. दिस इज कॉल्ड डबल ट्राइसोमिक कंडीशन. डबल ट्राइसोमिक कंडीशन मीन दो क्रोमोसोम ट्रिपल फॉर्म में मौजूद हैं इस वक्त. एग्जाम्पल ट्राइसोमी 21 डाउन सिंड्रोम. बेस्ट एग्जाम्पल लाइन-फिल्टर सिंड्रोम 27 XXY. XXY सुपर मेल कंडीशन. Tetrasomy. Tetrasomy results due to the addition of two chromosomes to diploid set of chromosome. जब diploid set of chromosome में जब दो chromosome ऐड करेंगे, the condition is called tetrasomy. 2n plus 2. 2n plus 2. Two types हैं. Simple tetrasomic, addition of two chromosome to one pair. Two chromosome to one pair, that is called simple tetrasomic. Simple जो normal diploid है 2n है, chromosome pair present in the pair. When in tetrasomic condition, the C chromosome is present in the टेट्राड 4 क्रोमोसोम इसके मौजूद हैं. दिस इज कॉल्ड द टेट्रासोमिक but simple tetrasomic condition. Double tetrasomic, 2 chromosomes are added each to 2 different chromosomes. 2n plus 2 plus 2. दो different chromosome पे 2 chromosome add होंगे. That is called double tetrasomic. Normal diploid 2n, only 3 pair of chromosome. When in case of tetrasomic condition 2n plus 2 plus 2 दो chromosome के साथ double chromosome है. B भी tetra form में मौजूद है और C भी tetra form में मौजूद है. This is called double tetrasomic condition. Examples of tetrasomy: Killian mosaic syndrome is a developmental disorder that affects many parts of the body. Most children with Killian mosaic syndrome have sparse hair on their heads, particularly around the temples. These areas may fill in as affected children get older. Cat eye syndrome, where a partial tetrasomy of chromosome 22 is present. Number third example, 48 chromosome, that is the tetra X chromosome syndrome is present, having the 4X chromosome. 2X2Y syndrome having 48 chromosome, and line filter chromosome having triple X and Y syndrome, line filter chromosome. These are the example of the tetrasomic condition. Diploid में E chromosome occurring pair. एनोप्लॉइडी नलीसोमी में पूरा क्रोमोसोम जो पेयर है वो खत्म हो गया. यहाँ पे अब 4 पेयर हैं, यहाँ पे अब 3 पेयर्स रह गए हैं नलीसोमी. मोनोसोमिक में 2n-1 सिर्फ एक पेयर क्रोमोसोम का डिलीट हुआ. मोनोसोमिक कंडीशन 2n-1. डबल मोनोसोमिक में 2 क्रोमोसोम, एक यहाँ से डिलीट हुआ, दूसरा यहाँ से डिलीट हुआ. डबल मोनोसोमिक. ट्राइसोमिक कंडीशन में एक क्रोमोसोम ऐड होगा यहाँ पे, और टेट्रासोमिक कंडीशन में 2 क्रोमोसोम ऐड हो रहे हैं, और डबल टेट्रासोमिक कंडीशन में 2 क्रोमोसोम यहाँ पे भी ऐड हो रहे हैं, 2 क्रोमोसोम यहाँ पे भी ऐड हो रहे हैं. This is the double tetrasomic condition, and this is all about the aneuploidy. नलीसोमी, hypoploidy and hyperploidy. हाइपोप्लॉइडी में मोनोसोमिक कंडीशन, नलीसोमिक कंडीशन, और जो हाइपरप्लॉइडी उसमें ट्राइसोमिक और टेट्रासोमिक कंडीशन, बोथ प्रेजेंट इन द सिंपल और डबल कंडीशंस. Mosaicism, that is the half of the cell have normal and remaining have abnormal in term of structure or number. जब some cell have the normal chromosome and some have the abnormal chromosome, both in case of the structure and number, their type of cell, their type of condition is called the mosaicism. It is commonly seen in Down syndrome or Turner syndrome. Significance of polyploidy play an important role in plant breeding and horticulture. इसका बहुत important role है, desired character produce करना चाहते हैं. Polyploidy has economical significance. It results in the production of large size flowers and fruits. Polyploidy play a significant role in the evolution of new species. There is a combination of different species. Each organism have a variety or the different number of the chromosomes as compared to their parents. Polyploidy results in the changes in the season of flowering and fruitings. Polyploids are vigorous invaders of new habitats. It leads to the formation of new varieties which show high resistance to disease and increase in yield. High जो new varieties produce हो रही है, जिससे resistance भी increase हो रही है और increase भी yield हो रही है, yield भी increase हो रही है. Tetraploids, cabbages and tomatoes contain more ascorbic acid, whereas tetraploids corn contain more vitamin A. Polyploidy varieties like apple, pear, grapes and watermelons are cultivated because of their large size. These are different significance of the polyploidies or the ploidy. That's all about the chromosomal aberration. Numerical chromosomal aberrations: the number of chromosomes deviated from the normal. It has mainly 2 types: aneuploidy and euploidy. Aneuploidy, hypoploidy and hyperploidy. Hypoploidy, monosomic condition, and nullisomic condition. Hyperploidy, trisomic or tetrasomic. Euploidy has 3 types: monoploidy, diploidy and polyploidy. Thank you so much.